And that experiment has already been done for mice right now. And there was a researcher in Boston named Rudy Yanish who took a mouse that had sickle cell anemia, where there was a defect in the gene, in the hemoglobin gene in the mouse bone marrow, and this mouse produces abnormal red blood cells. And in humans, this is a serious disease. In these mice, they collected the skin stem cells, reprogrammed them by putting in 4 new genes into iPS cells, genetically corrected the sickle cell gene in these abnormal iPS cells using something called homologous recombination. So they loop out the bad gene and put in a corrected gene. And then they could differentiate, give signals to make these iPS cells into bone marrow stem cells and do a bone marrow transplant and correct the hemoglobin abnormality, the red blood cell abnormality in the mouse. And so all this is feasible now, and the leap is to bring this to the right places in clinical practice. Okay. Another cool thing about stem cells is that they seem to know where to go. So, for instance, if we do an allogeneic bone marrow from a male who has an X chromosome and a Y chromosome in all their cells, and take those stem cells and put them into a female, and all the female cells have 2 X chromosomes and no Y chromosome, we can look for where the Y chromosome ends up, what tissues it's in, and then we know where the bone marrow has contributed to the growth, which organs the bone marrow has contributed to. And so, of course, when we do this and we look in female recipients, we see male cells form the blood, the immune system, and blood vessels, but also we can detect male cells, bone marrow-derived cells, in the brain, in the gut, in the skin, in the heart, in the liver, in the nail beds, and in the nails. So a lot of different tissues are derived from bone marrow stem cells. And there's this thought that when you take a cell that has a stem-like property and transplant it into a patient, that stem cell can travel around, and if it lands in the right environment, it will take clues from that environment and differentiate and help form that tissue. Now, right now, we can do bone marrow transplants and make the whole blood system of some of the recipient the same as it was from the donor, but only about 1 or 2% of these other tissues would be derived from the bone marrow. And so it's not a practical technique right now to repair organ damage, for instance, to repair heart attacks or to repair strokes using this technique. But as we understand how the stem cells do this, we can then augment and make these techniques better. And so the hope is in the not too distant future that we can use stem cells to repair injury to these different organs. There's lots of different ways that we can manipulate stem cells. And so if we look at stem cell therapy right now, what we can do, we can purify them, we can take them out of the body, and we can expand them in tissue culture. We can genetically modify the stem cells, either to correct a defect or to make these stem cells produce something that will make them better. We can kill stem cells, for instance, cancer stem cells that might be leading to making a patient sick. We can kill those with chemotherapy. We can stimulate the stem cell growth in the patient's body using agents we call cytokines. We can instruct these stem cells to differentiate either in the body or outside the body. And we can produce scaffolds that then help direct the structure of the stem cells into forming new tissue. And so individually, all these techniques have been developed over the last 5 years, and we're hoping in the next decade to bring several of these into medical practice. And so, for instance, right now we're doing a clinical trial to see if this is worthwhile for a patient, who, for instance, who has a heart attack and comes in with chest pain and damage to the heart because one of the blood vessels has been blocked, and it cuts off the supply of oxygen and nutrients to the heart. That heart muscle would die. But our thoughts are, if we give this drug called GCSF, or granulocyte colony stimulating factor, or Neupogen, to the patient at the time they come in with their heart attack, it mobilizes the stem cells from the bone marrow into the circulation. And perhaps by allowing these stem cells to circulate, they'll be captured at the damaged area of the heart and help it repair. And preliminary tests in humans have suggested that this is the case, and we're doing the trial right now to see if this is true or not. And so you can see, so these are scans where they're called false color scans, and they look at metabolic activity. And so where the heart muscle picks up a lot of dye and has a lot of metabolic activity, meaning it's functioning normal, the heart scan is red. And as the activity diminishes, the scans go from yellow to green to blue. And so this is a region where there's no blood flow and no metabolic activity and no pumping. And so this patient would have a lot of problems. After the treatment with the Neupogen, you can see this area has started to fill in. So this is the same picture of the heart. And so there's some recovery of the heart muscle, and so this patient would have less disability, less damage to his heart afterwards. And so we're hoping with our preliminary analysis holds up in further testing. Now I want to switch to multiple sclerosis and tell you about a project that we're doing at the Ottawa Hospital right now. So multiple sclerosis is an autoimmune disease. Normally what happens is our immune systems circulate around our body, and they recognize what's a germ when it comes into our body and attacks the germ but leaves our body alone. But sometimes the immune system gets miseducated and confused, and what it happens is that it learns that part of our body, or it thinks that part of our body is like an invading germ and starts to attack it. And in multiple sclerosis, that attack happens on a cell called the oligodendrocyte. And these are insulator cells that wrap around the long wires of nerve cells. These long wires are called axons. And so when the immune system attacks these oligodendrocytes, it prevents nerve conduction along the nerve cells, and as well it kills the nerve cell, because the nerve cells rely on these oligodendrocytes for trophic factors to help them survive. And that produces disabilities in patients. And we can see evidence of this disability when we examine patients, but we can see evidence of the damage on brain scans. And so this is a normal brain scan, and these white areas are areas that have been damaged by attacks of the immune system.